Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and this is the third and the final live sessions. And so, if you have any question, you can ask me. Sudarshan, how are you? Sudarshan, you can turn on your microphone and camera also, so that you can interact with me. Sudarshan, are you getting my voice right now?
Yes, sir. Hello.
Yes. Can you turn on your
Yes, sir. Good.
camera?
Sir, the camera is giving me a bit of a problem, sir. Only microphone is work right, sir.
Okay. Okay.
Yeah. Yes, sir.
Uh,
Only yeah. Yes, sir. Yeah.
okay. So, can you introduce
Yes.
yourself?
Yes, sir. I am Sudarshan Gohai. I am I am one of the participants in this course. Yes, sir. I am
Hmm.
from Guwahati itself. I am from Guwahati only. Yes, sir.
Okay, which college?
Sir, I am actually working. I was working.
Oh, you're
Yeah.
working.
I, yes, sir. Yes,
So
sir.
what is your qualification?
Sir, I have done my BSc in in physics.
Okay.
Yes, sir. Yes, sir. I
So
actually I have picked up this course just to know a little bit more about the subject. That was my intention. Yes, sir.
okay. So did you find the
I
subject?
do not. Yes, sir. Yes, sir. Very much. I found it very interesting, and thank you, sir, for giving us the opportunity to be part of this course.
So yeah. So do you have any query? Specific query?
Yes, sir. Yes, sir. Sir, simple because my I'm it's a simple head, sir. Sir, when we in the context of the cell cycle,
Mm-hmm.
yeah, when cells are entered the G naught phase, then we
Mm-hmm.
say that they are metabolically active, but they no longer grow. So, sir,
Mm-hmm.
I know this simple question, but I'm not from bio background, so I don't understand this part. That we say that they are metabolically active, but they can no longer grow. I'm not very, and I don't understand this part, sir.
Okay. It's very simple.
Yes, yes,
Okay.
sir. Yes, sir.
Like like for example, myself. Okay, I will be in G0
Yes, sir.
phase right now because I am
Yes, sir.
metabolically active, but I don't grow. And I may be growing because I am replenishing, whatever I am losing. For example, every day I am losing some part of my skin, I am losing my hairs, I am losing many things,
Yes,
right?
yes,
And
sir.
all these are actually going to be synthesized, right?
Mm-hmm.
But I'm not growing, right? My height is not increasing. So that's a way. But this is just a very crude example. The simple example is that metabolically active means the cell is taking up the nutrition from the body, and it is
Mm.
maintaining the equilibrium. Like whatever the molecule it is degrading, it is synthesizing the new molecule. Why it is not dividing? Because it's so growth means it is maintaining the shape and size, but it is not dividing. It is not dividing in terms of to increase its number.
Yes, sir. Yes, sir.
Any
So
other
it is not
specific
dividing.
question?
It is not dividing, but it is taking nutrition and carrying on other processes.
Yeah, it is maintaining whatever is required to maintain the life.
Right, sir. Right, sir. I understand, sir.
Just like when we do the same thing, we achieve, when we reach to the maturity, that time after
Yes, sir.
that we just maintain our, for maintenance purpose only we take the nutrition.
Yes, sir. Yes, sir. Sir, in one of the questions in one of the assignments, it was, I think, some cells of the retina of the human eye, they do not grow
Mhm.
after a certain point. Is that correct, sir?
I exactly don't remember the assignments because until
Yeah,
I
yes,
will
sir.
not see the question. I think it is difficult to comment.
Uh, I think
If
it was
you
said something endocrine cells, sir. Endocrine cells of the retina.
Okay,
Yes, sir. So one of the options was that, and that was the correct option. It seems that those are the cells which do not grow after a point.
mhm.
Yes, sir.
Okay, okay, yeah.
Yes, sir. Yes. Okay, sir.
Atia,
Yes, sir. Hello.
okay. Can you introduce yourself?
Yes, sir. My name is Atiya Sheikh and I'm from Pune.
Mhm.
Sir, my question is like that. Sir,
What is your
exam
background?
is hello.
What is your background?
Sir, I am BSc student. I am
BSc what?
currently studying in BSc, third year BSc.
BSc biology.
Yes, sir. BSc microbiology.
BSc microbiology. Okay, so
Yes, sir.
in Pune University you have pure microbiology. Means like
Yes,
you
sir.
don't have other subjects like chemistry and other thing.
No, sir. We have other subjects like chemistry, micro, but I chosen microbiology.
Okay.
Yes, sir.
Okay, you can ask your question now.
Yes, sir. My question is like that. The exam is going on to 23rd or 24 April. Okay, sir.
Mm-hmm.
Oh, sir, when we will get certificates of our exam?
When we will get our certificate? I don't know.
ये
Maybe
exam
sometime. I think sometime in May. It doesn't
May.
take
Okay,
time.
sir.
It doesn't take time. Maybe 2 weeks.
Okay, sir.
Exact date
Cause
I don't.
yes, sir. Cause our teacher have said us that they have to update our credits. So I asked this question to you.
Hmm, hmm,
Then when
hmm.
will we get certificate? Yes, sir.
I don't know exactly because that date is anyway not mentioned in the course. But um, as far I remember, I think it doesn't take more than 2 weeks
Okay,
for
sir.
me because within suppose the exam will be done on 33rd. Within a week, I think I get the transcripts. Within a week, I get the corrected copies, and then I do the grading within a week or so.
Okay, sir. And sir, sir, our certificate will be in online mode. Yes, sir.
Yeah, yeah, you will get a soft copy.
Okay, sir. Thank you, sir. This is my question. And the assignments are so awesome, sir. I have solved it. It was
Mm.
very nice. And your lectures are so beneficial for us, for me
Mm-hmm.
also, in my
Mm-hmm.
microbiology studies. Yes, sir.
Mm. So you don't have any other specific question related to subject?
No, sir. No, sir. If I have a question, I'll ask you.
Yes. Okay. So
Yes, sir.
let me
Can we
share my
leave or we have to stay?
uh, if you want to listen me, you can stay. Otherwise, you can leave.
Okay, sir. Okay, sir.
Because I want to share one because one of the in the discussion forum, one of the students were asking about the
Yes, sir.
Hmm.
nerve conduction,
Yes, sir. Yes, sir. Yes.
and I said that I will discuss about that because there's some confusion.
Yes, sir.
So
Yes.
yeah. So Sudarshan, I have just mute your microphone because otherwise it will give a background noise. Okay. For Atiya also I'll do the same. Okay. Otherwise it will make an echo. So I'm sharing my screen and okay. So if you recall from last 3 to 4 semesters, 3 to 4 modules, I think we were mostly discussing about the physiology. Okay, and what we have discussed about most of the major physiology system, right? We discussed about the digestion, circulation, muscular, nervous system, endocrine, excretory, and in the module 11 and 12, we have also discussed about the homeostasis as well. And one of the important aspect is about the nerve conductions. Okay, so nerve conduction, I think if you see this particular slide, the confusion, I don't know from where we got the confusion, but the nerve conduction is a 3-step process, right? In the step 1, you are going to be, all the nerves are going to be on a resting phase, right? And in the step 2, they are actually going to receive the signal, so they will be under the action potential. And once the action potential is over, then it has to be go through with the repolarization. Okay, so all these 3 events are actually being governed simply by the exchange of the ions. Okay, so if you have with the exchange of the ions, you can be able to keep changing the polarity of the membrane. Okay, it means, for example, under the resting stage, you are going to have the negatively charged inside and positively charged outside. Okay, and how you're going to maintain the negatively charged inside means you are actually have to hello. Yeah, so how you're going to maintain a negatively charged membrane, that you are actually going to throw more number of positively charged ions outside than the amount of positively charged ions what you're going to inside. Okay, so that's how you are you are actually going to maintain a negative charge inside. Now, you can imagine that when there will be an impulse, right? When you are actually going to have the impulse. So in this case, for example, when you are in a resting stage, the more number of potassium are actually going to be thrown out, and less number of sodium are actually going to be taken up by the sodium potassium pump. And as a result, what will happen is that there will be a negative charge throughout the inner side of the membrane. Now, once you with the neuron is actually going to get a signal, some of the ion channels are actually going to open, and because the ion channels are going to be open, especially the potassium ion channel, the more number of potassium are actually going to enter inside, and because of that, the negatively charged ions are actually going to be replaced by the positive charge. Okay, and that's how you can imagine that at point number 1, for example, this is the point number 1, the negative ion is actually going to be replaced by the positive ion, and that's how this is going to be changed. Now, once this is going to be changed, the signal will move. Okay, and then it will reach to the point number 2. At point number 2, again, this is going to be changed and it is going to be converted into positive ion. And that's how the signal will vary from 1 to 10. For example, if you have the 10 points, and this is the neuron, right? The signal is actually going to originate here, right? And it will travel all the way up to here. Okay. And while it is traveling, and if there is no stimulus, then this part of the neurons are actually going to enter into the repolarization, and under the repolarization again the same thing will happen. The sum of the protons, potassium are actually going to be thrown out, and that's how the point number 1 is again going to be revert back to the negative. Then point number 2 is again going to be revert back to the negative, like that. Okay, so it's going to be positive, it's going to get converted into negative. And that's how you are actually going to relay the nerve impulse from one place to another place. And ultimately, once the impulse is over, then you are actually going to revert back to the original state. So I'm sure with this brief discussion, probably that particular type of confusion could have been cleared, right? But I will suggest, even if you have some more queries, you can still ask me, because we have very limited time. We have very limited time. Yeah. So we have very limited time, so that we can actually be able to discuss. Sudarshan, if you don't have any, you can leave also if you want to.
Yes, sir. I do not have any more queries from my side. So thank you, sir, for this opportunity which you have given us for this class.
Yeah, since let me tell you one thing. Okay, if you are
Yes,
more
sir.
interested in exploring more about the biology,
Yes, sir.
and if you want to have some hands-on sessions or something, uh, uh, try to join some workshop or something, because then only you will be able to appreciate the biology part. Otherwise, reading only the theory part doesn't make much sense. When you I'm sure you have never seen the DNA, right? By your naked
No.
eyes. Okay, so when you see the DNA with your naked eye, you will be very excited about that.
Sir, where can I join
have you
this?
seen a mammalian cell?
No, sir. No, sir.
No, you have.
No,
Have
I
you
have
seen
I
the
have not
mammalian
seen.
cell?
No, sir. I have not
You
seen it.
have.
Sir.
I'm 100% sure.
Yeah.
What do you eat in your breakfast?
Oh, yes, yes. That way I have seen. Yes, right. That way I have seen, sir. Eggs and all. Yeah, yeah.
Yeah, egg is
Yes.
the one of the mammalian cell only. Okay, and it's a single
Right,
cell,
sir. Right.
single cell. If you take the
Yes, sir.
fertilized egg, it is actually a cell where, like for example, in our lab, we normally use those kind of eggs for growing the virus. So when you see a very virus grows, you will see very nicely. You can be able to see all the membranes and all the organelles. You can be able to see that.
Right, sir. Yes, sir.
Leelavati, can you introduce yourself? If you don't have any more questions, we'll wind up actually the live sessions. So if you have any questions, for example, like we have Leelavati, I think if she has any questions, probably I'll be happy to answer.
Hello, sir.
Yeah.
Yes, sir. My question is that the major force that stabilize a double-stranded DNA in aqueous solution. So sir, hydrogen bonding or hydrophilic interaction, which was there, sir?
Can you repeat?
The major force that stabilizes a double-stranded DNA in an aqueous solution.
Mm-hmm.
Sir, then which force which stabilizes the DNA?
It's you're asking from assignment, right?
Yes, sir. My answer is wrong in this assignment.
So what was your answer?
My answer is like I choose my answer hydrogen bonding, but it's saying that base stacking. Accepted answer.
Hmm. Hydrogen bonding is very weak. See, when you have when you have an objective question, it is not that hydrogen bonding is wrong answer. Okay,
Okay, sir.
but it
Yes, sir.
is relatively less accurate
Okay, okay.
because base stacking is actually going to be very strong force.
Okay, sir. Base stacking.
So when we say stabilizing the structure, we have to go with the major force, what is responsible for that, not the minor force. So I will not say hydrogen bonding is not essential. Hydrogen bonding is also playing a crucial role. For example, if you just change the pH,
Yes, sir.
if you change the pH, you are actually going to destabilize the DNA structure.
Okay, sir. I
But
understand.
that time you are actually changing the you're only playing with the hydrogen bonding. But when we
Mm-hmm.
say in under the physiological condition, under the water, which means we are talking about the normal pH, the waste stacking is actually going to be more crucial and more important and more strong force compared to
Oh,
the hydrogen bonding.
okay, sir. And sir, my question is one of my question. Sir, in criteria to get how to get certificate. So, sir, there is an criteria like that 25% of assignment score, and how many percent we have to score in the exam. I didn't understand that
40,
criteria.
I think I exactly don't remember, but I think 40%. If you
40%.
if you
Okay.
score less than 40%, you will not get a certificate.
Yes. So, sir, I didn't understand that criteria, so I ask you.
I think 40%. I exactly don't remember, but I think it is 40%. What is written on the instructions?
Okay, sir. Thank you, sir.
Okay, good. So if you all don't have any question, and I only have 3 students for interactions, I will wait for 1 more minute. If we don't have, then I think we'll wind up this live session. Leelavati has joined, but I think either she is not getting my voice or I don't know why she is not responding. So Sudarshan, what job you do actually in Guwahati?
Hello, sir.
Yeah, what you're doing?
Actually, sir, I was teaching, but
Oh,
right now, because of family issues, I am at
hmm,
home taking care of my of one family member who is not well, sir. That is it.
okay. So where you
Yeah.
were teaching before?
I was teaching at industrial mass communication, sir. Mass communication.
Okay, okay,
Yes, yes,
good.
yes, yes.
Okay, Sudarshan, Tia Nilavati, I think we had wonderful interactions. It seems none of the students have any more questions, so I'll wind up the live session now. And thanks for your joining the live session.
Thank you, sir.
And I hope the course will actually give you at least some idea about the biology. This course is a
Yes,
very
sir.
basic course. This course is only for the non-biology student mainly. So that's why I try to keep it very simple. And at the end, there was little complications, and it was little advanced. But overall, it is from my side. I think I have tried to make it as simple as possible. So with this, I think I would like to wind up the live sessions, and I hope you will do very good in your NSEM exam. Uh, Definitely, even if you have done all the 11 assignments, you should definitely attempt the assignment 12, because that will give you an idea what kind of exam, what kind of questions you are going to get in the NSEM exam. So with
Okay,
this, all the best for your NSEM exam, and I'll wind up the live session now. Thank you.
Thank you, sir.
thank you.
Thank you so much.
Thank you, sir.
Thank you, sir.